I'm glad to see you, my friend. We're talking all things yeast genetics today. We are very slowly dissecting this video. This guy won the Lifetime Achievement Award for studying yeast and yeast genetics. And so we've just been kind of chatting about. I've been pretty much pausing it at every slide just to talk about some of the concepts behind what they're talking about. But it's, I think, really cool to what all you can use yeast cells to study for. And I think whoever's interested in the talk, I will also upload it onto the Discord if y'all are interested in seeing it. I like downloaded a lower res format, so it's a smaller file size, so y'all can watch it if you're interested. We're doing part 3 of our yeast genetics deep dive today. Remember, last week we did a series of experiments where we looked at yeast grains, yeast genetics. We did this experiment last week where we took yeast granules. This is just baker's yeast. Theo, who is talking about baking as well, will probably have some in their kitchen. We looked at them under the microscope like this. This is what they look like, the little packets that you have in your kitchen. And then we hydrated them the same way. And then we actually saw cell division occurring underneath the microscope last week. We did this exact same experiment. I'm just doing a little refresh on YouTube so we can see it, like how our results looked as well. And it was super cool to look at and see the cell divisions. We actually saw all the buds forming. And that's one of the main reasons why yeast are an amazing organism to study, is because they're more simple than humans. And a lot of the processes that they have, other cell types have too, like cell division. Reviewing the experiments that we did last week on last Saturday, which was rehydrating yeast, baker's yeast, under the microscope. And we got to see live cell division underneath our scope. And this is just a summary video of exactly the experiment we did here on stream. Where after you rehydrated the baker's yeast, you saw this effect happening, which is, we actually saw live cell division. We saw the buds forming from all the yeast molecules. And that's actually the inspiration for our budding yeast character as well, right? There's bud and buddy, and you see this little bud form. And it's the same way we're seeing here too structurally. There's a smaller bud coming off, and this is just mitosis. So rudimentary cell division between these organisms, which again, it's just really cool to be able to take something quote unquote as simple as baker's yeast and see just like cell division happening in real time, right? I think that's really remarkable and really exciting to learn about and see. And so yeah, I just wanted to show that off. This is just kind of the primer. I have this talk that we've been kind of chatting about for a few weeks now. And I'm excited to show it to y'all because it is a Lifetime Achievement Award in yeast genetics and all that this guy did. And there's a really nice introduction for who he is. And the speaker was a graduate student, which was really cool to introduce him. And then he kind of goes into his lifetime of work in a very accessible kind of way. So I really like that. The Yeast Lifetime Achievement Award. So this year it went to this gentleman, Dr. Tom Peets, who, again, we're just going to kind of talk about his presentation, and his is about how genes and the genome is regulated in budding yeast, and that's what his work was on. So we'll pause it probably a fair bit to talk about some of the science. If anyone has any questions during it too, just pause it and we'll rabbit hole. So basically what he's researching is how quickly cells divide and how that affects mutation rate. So very fundamental genetics for it. And then if you stress out the cells during replication, what happens to the DNA? Crossing over. What this means, y'all, actually it's really nicely illustrated in these colors. Wow, it's hilarious. If I tried to zoom in, if I zoom out, it gets bigger. That makes total sense. So these, if you view the blues as chromosomes, when they're dividing, there's the possibility for there to be a switch between the blue and the red. So instead of having just the red chromosome come out and stay red and the blue chromosome come out and stay blue, there is the effect possible of them mixing, so that the blue now has a little bit of red and the red has a little bit of blue, and that causes genetic variation. And so how does that happen all the time? Is one question that can be answered and studied in yeast, which I think is really cool. Are they looking to find ways to do that intentionally? So, Chris, they are trying to. So, the research was done to identify how this happens and how to get it to not happen, and then how to get it happen in a targeted way. So, basically, to fully understand the cell's machinery, what you need to do. So, this is a common term in genetics: necessary and sufficient. What that means is, if you find a gene that you think is responsible for something, you have to show that it's necessary for the thing you're observing. So if you get rid of the gene, okay, it will no longer happen. And then 2, sufficient, meaning if you turn on that gene by itself, this thing that you want will happen. So those 2 components, necessary and sufficient, are what you need to really show what the genetic point of something is, if that makes sense. That's the goal, Chris, is to figure out, okay, how does it normally happen under, like, in the genetics world? Like, what genes are involved in this? Because this process happens in us too, right? You get a chromosome from mom and a chromosome from dad, but when they're splitting, like together in the cell, these crossover events can happen. So you can have mixes, and these recombination events is what make us look not like a clone. So if you can study it in something more quote-unquote simple, like a yeast cell, and understand how it works, it's easier to then understand how it happens in more complex organisms. And even, not necessarily directly to humans, but to a fruit fly or to a fungus, and see like if that's still the same process. Turns out it is. The genes are conserved between these organisms and what they do. It's just you add more complexity as you move up in complexity of species too. I think these are fundamental biological concepts that are kind of cool to talk about. So they're diploid, meaning they have 2 copies of every chromosome. Haploid is single copy. Haploid, you're not going to have crossing over because there's only 1 copy of a chromosome. Diploid, you need the 2 copies, and then you'll start having this crossing over events. Next level genome project. Yeah, Chris. I mean, Chris, they've sequenced the genome in humans and many, many organisms because it's becoming much cheaper now as well. But there's still, like, how to manipulate the genes is a very, very new field. We were talking about crossing over events with the chromosomal switching back and forth, just because that could cause a disease state, such as retinoblastoma. So a crossing over event can lead to a negative outcome where you can lose certain genes, and that loss will cause negative effects. Like you can see one eye in this patient is normal, and the other eye is not, because the other eye lost a tumor suppressor versus the normal one didn't. And that can lead to blindness in one eye but not in the other. So there are genetic issues to always consider. So this eye is the homozygous for the mutant, and this one isn't. So that's why you can actually have like a chimera basically inside of a human. These cells are abnormal and these are normal. Then now with this crossing over event, what ends up happening is 3 of the 4 chromosomes might have that red gene. So the 2 red original ones, and then this crossover events where it adds in a little bit of the red, and that could be a good or a bad thing. Depending on what gene has that crossing over event, where it comes from one chromosome and goes on to another that didn't have that particular copy of that gene. It could be good or bad. Just saying, there's different ways that these crossing over events can happen depending on what the shape of the chromosome is. So sometimes if the chromosomes end up looping, then you can have a whole rearrangement of how the chromosomes are organized, or the genes on a particular chromosome, and that directionality. So A going to B going to C, if it's shifted around that B now is facing in a different direction, that could be a really extra negative thing. Because it could be when a promoter lands on a gene and turns on that gene, it needs to continue going to turn on the second gene. But if the chromosome is in a reverse orientation, it does not work. appropriately. So there's all these extra things to keep an eye out for when you're undergoing recombination. Gene recombination events, meaning to identify the individual components of a gene and how they recombine and how they can inform us of additional, like, new things, like how cells connect, how the chromosomes are divide, how there's genetic shift from one organism to the other. And there's so many combinations that it could happen, so there could be a lot of negative things that happen with these crossing over. So remember, it's like, think of this blue chromosome and this red chromosome. They're both chromosome 1, let's say. One is from mom, one is from dad. But when they have this crossing over, they're exchanging genetic information. That could yield a couple of things. One is extra copies of one gene on one chromosome, less copies on another chromosome, or just switching the genes between them. And that can yield a bunch of different effects on the physical state of of like a human or any organism, which I think is really neat. So sometimes there are recombination events that you can't readily see, but it's kind of, so it's less likely to have a physical effect, and it's just harder to sequence. So you have to do different techniques to actually identify them. This is just a very clever way that he did these experiments. That I was really excited about how elegant this was. So they have, again, it's the red and the blue chromosomes, and he's trying to measure how often there's a crossover event, and he can actually measure that visibly. So you can see here, these yeast cells that didn't have a crossover event stay their original color, but then when there is a crossover event, you get this little influx of red and white. So you can actually see when there are these changes that take place. And so then you can actually count them and do the mathematical modeling of how often recombination takes place and why that matters and what downstream effects it has. Meaning, like, if I make the genetic changes here, what effect will come of it? I think it's super cool that you can see this. And you can also map the distances. So right here, that what he wanted to ask was these two genes, like how often do they recombine, knowing like where they are in the chromosome even. And you can actually visually identify cells that have undergone recombination, which is super, super cool by just this red stain on the cell. So the ones that are red have had a recombination event, and the ones that aren't have not. And so you can actually sift through them and pick out little colonies on the plate that you're interested in studying just by color, that you can actually see. You don't need any fancy pants equipment for it. So you can actually map and see how chromosomes have changed, how they've had crossover events, and what's done the crossover by this color. So again, these are genes that they put in to give these colors, and then they can just see how it works and like how the crossover events happen. Double question: approximately how many generations of yeast will there be in a day? And is this one of the reasons it's so handy to study them like this? So Chris, it's temperature dependent. Higher temperatures to a certain point allow there to be more generations. There can be a whole doubling event in about an hour at 37 degrees Celsius. So you can go pretty rapidly. You need to make sure, if you want a full 24 for a single one, like of divisions for one particular one, you also have to make sure there's enough food. Like the substrate that you're growing them on has to be like pretty nutrient rich. And that's yes, it is one of the reasons why it's good to study them. Because if you're studying questions of cell division, because you can make them divide pretty readily, and they also have a smaller genome, that's what makes them handy to study. Because you might be like, well, Belent, why don't we just study human cells? There's far more chromosomes and just total genetic information in the human cell. And remember, these have been studied since like the 1920s for yeast. So it has been an organism of study for a very long time. And so figuring out the fundamentals of them, even before we had the fancy technology, just made more sense, both from an economical perspective of cost of experiments, but also from just ease. Yeah, exactly, because just smaller genome size. And it's, so obviously there's then a ton of genes that they don't have, that humans do have. And so that added layer of complexity is just, is important to note, but you can't study the complexity if you don't know what the fundamentals are. And so using an organism like this to study the fundamentals and then move on to something more complex, I think is a good way of doing things, in my opinion. So there's a lot of jargon here, which I like to, this is like a good way of, I think just like going through some of the fundamentals on this. So you sort out the fundamentals first, learn to crawl. Exactly, Chris, exactly. So another way to visualize these kinds of chromosomal changes are called genetic markers. When you put in a gene that gives you something visible to see. So, for example, this particular yeast cell can have this genetic marker called W303A. And that, let's just say, makes it red. And so if anything's have a crossover, but it, like that gene that makes it red comes over, then you'll be able to see if it's recombined or not, because it's either lost the gene to make it red or it's gained a gene to make it red. So you can see if it's happened. Same thing with this other marker, this YJM789 that turns it blue. Same kind of question. And then if you mate them, they might not have either color, they might be a third color. So you can actually track, okay, which cell type is haploid, meaning only have one copy of every chromosome. That's like the red and the blue here. Diploid, meaning having two copies of every chromosome. That's the black here. And it's all through these visible markers. You can also do ones that's like a growth-based experiment. So you take these cells that have these markers, and the marker might not be a color thing, but rather if it can survive on a dish with antibiotics. So does this marker give it antibiotic resistance? So that's one thing you can also do instead of, again, a visible color, is just seeing if it grows on a dish. And let's say you put antibiotics on a dish, and only the ones that have a certain gene can survive on the antibiotic. And so you can actually then count, okay, how often does the antibiotic gene get lost or recombined in? You can ask questions like that. And again, because they divide so much, you can ask like colony growth. Like Chris, on an agar plate about this big, if you streak a colony, meaning if you take like a little stick and you draw a couple of lines on the plate, that whole dish, if there's nothing like an antibiotic or something that's like testing for growth, that entire dish will be covered by the next day with little colonies of yeast. So it grows very, very rapidly. Same thing with E. coli as well. So I like this component of the talk because he's basically admitting like, okay, this can be complex. If you wanted to see if there was changes in the genome, you could sequence it, which is what we could do nowadays, or he's giving the alternative approach of a microarray. A microarray basically was a plate that you could get, and on the plate there were a bunch of little strings of certain sequences. And then what you did was you took your genome, in this case the yeast cell, and you saw if its DNA stuck to any one of those little sequences on the plate. When it stuck to a sequence, it lit up. And so you can not only measure what sequences were most common in your pool, but also how often the crossover event happened. So like if there was more glow of one particular sequence, you know that was much more prevalent in your population than one that had far fewer glow or no glow at all. And again, so it's just a simple binding. It's taking a known DNA sequence that's stuck on a plate and seeing if your DNA sequence will bind to it. And if it does, it glows. If it doesn't bind, it doesn't glow. And that's like the easy way of doing it. You can actually buy microarrays, like these little squares, and just like run the experiment and see what glows and what didn't. Those also were not super cheap back in the day, but now they're very inexpensive. Sequencing has also become far more inexpensive, but a lot of it's coming down in price. But still, the old school methods I think are really neat.
Thanks for watching SciAnts Media, and be sure to hit those like and subscribe buttons to help our channel grow and support our science outreach and education goals. We can be found on Twitch most days of the week, and be sure to join us here for our next video. Thanks again, and stay curious, everyone.